Today, I will be presenting on peptinase enzyme for paper recycling. Paper recycling depends on the inking, removing ink from printed paper waste so fibers can be reused. Traditionally, this is done with harsh chemicals like sodium hydroxide and surfactants. These chemicals strip ink effectively, but they also pollute water and damage ecosystems. Ink particles cling to paper fibers because of peptin, a glue-like substance in the paper matrix. Peptinase breaks down this peptin, loosening the bond between ink and fiber. That is why the inking needs peptinase. It allows ink to detach naturally without toxic chemicals. This research is important because it solves several problems at once. Environmentally, it reduces water pollution by replacing chemical de-inking with a cleaner enzyme-based process. From a waste management perspective, it gives orange peel, a common byproduct of the juice industry, a valuable second life. Industrially, producing enzymes from waste lowers costs compared to buying commercial ones, making bio-de-inking more practical for large-scale use. Scientifically, it shows how optimization methods can drastically boost enzyme yields. The enzyme was produced using submerged fermentation, where orange peel waste was mixed into a liquid broth and used as food for the Aspergillus niger. In this process, the fungus grows inside the liquid and release peptinase into the medium. Much like how microbes produce useful products in food fermentations such as soy sauce, the enzyme solution collected directly from the fermentation was then tested for paper recycling. To make production more efficient, the researchers applied Taguchi statistical design to optimize conditions such as pH, inoculum size, pH concentration, and incubation time. The best recipe was pH 6.5, inoculum 17.5%, pH concentration 2.5%, and 24 hours incubation. Among these, pH was the most critical factor, accounting for more than half of the variation in yield. This optimization increased enzyme yield more than fourfold, from about 200 to nearly 900 units per milliliter. The optimized peptinase was then applied in paper recycling. By breaking down peptin in paper fibers, the enzyme loosened ink particles so they could be washed away. In tests on newspapers and magazines, this method removed over 80% of ink, proving that bio-de-inking can be both effective and eco-friendly. Imagine cleaner, recycled notebooks in classrooms, produced without polluting chemicals. That is the promise of this innovation. Practical results that people can see and use. Challenges remain before this innovation can move beyond the laboratory. Orange peels vary in peptin content depending on source and season, which affects enzyme yield. The enzyme also did not always remove ink equally well from all paper types, so broader trials are needed. And most importantly, the study proved success only at lab scale. Scaling up to industrial recycling plants requires more testing, investment, and consistency. If these hurdles can be overcome, orange peel enzymes may one day transform paper recycling into a truly sustainable process, turning waste into value and pollution into progress.